Okay, welcome. Let's discuss about the food test. That is a practical area. That's a paper there in biology. We have chemical tests, and let's start with the food sample, which is the starch. What are we using? We're using iodine test. The procedure should be very correct. You put 2 ml of the food substance that you are given, add 2 drops of iodine, and we are very specific, 2 drops of iodine solution to the food sample. Your observation, if the starch is present, you'll check about the color change to blue-black, and you can check this one is blue-black in color, and it's moving from this way because the iodine solution is brown in color, and it will change to blue-black, and you can check it's blue-black. In case it's absent, you can check the color of iodine solution is retained, and we're saying iodine solution retains its brown color. Which are the key marking areas? You should show the amount used, which is 2 drops, and the word solution should be there for you to qualify a mark. And the marking goes this way. If you miss the procedure, you'll not get the observation, and you'll not get the mark on conclusion. That is it. That is the first one. Let's check about the second one, which is the reducing sugar test. What are we doing? The food substances are reducing sugars, all right? The test is the Benedict's test. We test for the reducing sugars. Let's have a very good procedure. You put 2 ml of that food substance that you're given, add 2 ml of Benedict solution to the food sample, then you eat to boil. So the concentration or the amount required is 2 ml. The 2 ml of the Benedict solution still. The Benedict's check how it's written, capital B apostrophe S, and it should be in solution, and that cannot still be a mark until you speak about eating to boil. From that point now, you can check about the traces of the reducing sugars present, and you can check about the presence of the reducing sugars. The traces you can check about this color changing from blue to green. So the blue is the color of the Benedict solution. If they're just in traces of the reducing sugar, you can check the color is green. And in case it's present in high amounts, you can check after you boil, it will first change to green, yellow, then brown or orange, but we are not red. So we are speaking about the color change. This is the alignment, green, yellow to brown or red, and not red, but orange. So what are we speaking here? You write the final color, and we're speaking about the reducing sugars present. What about if the reducing sugars are absent? You can check the blue color of the Benedict solution will persist. You can check that. Reducing sugar in your conclusion, absent. Number 3, let's check about the longest procedure in the food test. That is for the non-reducing sugars. Still, we do Benedict's test for the non-reducing sugars. So the first one is the procedure. You put 2 ml of the food substance, add 4 drops. First speak 4, or you speak about 3 drops of dilute hydrochloric acid. It's only hydrochloric acid that is not in solution form. All the other reagents are in solution form. Then you eat to boil, cool, add sodium hydrogen carbonate solution until fizzing stop. Add equal amounts or 2 ml of the Benedict solution, then you eat to boil. I think that is very clear. We are saying 2 ml of the food substance that you are given. You add at least 4 drops using your dropper of HCl, dilute hydrochloric acid. You eat to boil, then you cool. So hydrochloric acid is for the hydrolysis. Adding sodium hydrogen carbonate solution until fizzing is to neutralize the acid. Then add equal amount. So this one was first to break the complex non-reducing sugar to at least a reducing sugar. After that, now having the reducing sugar, you can do Benedict's test, of which you first add Benedict solution. That is a reducing sugar. Then you confirm after it has boiled, and the observation will just be like for the reducing sugars from blue Benedict solution to green, traces of the non-reducing sugars. Then from blue, green, yellow then to orange indicates that we have more of the non-reducing sugar test. In case of the absence of the non-reducing sugars, what happens? The blue color of the Benedict's persists. Then we have the purate test. Purate test, that is the test for the proteins. The procedure is very simple. You just put 2 ml of the food substance, add equal amount of sodium hydroxide solution, for the food sample, add copper 2 sulfate solution, and it's in drop wise, and you shake. So we have said you put 2 ml of the food substance, add 2 ml of sodium hydroxide solution, add copper 2 sulfate solution, drop wise, and you shake. So what happens in case it's present? We speak about the purple or violet color changing from blue to either purple or violet, and it will indicate the proteins being present. So we are saying this is a blue color of copper 2 sulfate, and it will change in case of presence to either violet or purple in color. In case it's absent, that blue color of the copper 2 sulfate solution is retained or persist, proteins will be absent. The second last, we have the ascorbic acid, and that is the vitamin C. Ascorbic acid, other name is vitamin C, and not just a vitamin. Add food sample to the DCPIP, that is dichlorophenol indophenol solution, dropwise, and shake, then observe. So here we start with the what? Solution. You add first what? Food sample to the DCPIP. So the DCPIP is what you measure 2 ml. So we are speaking here, you measure the 2 ml of DCPIP. Then now you add your food samples dropwise. The food sample is added dropwise, of which then you shake, of which what will you observe at the moment? The DCPIP will get decolorized. It will get decolorized. So ascorbic acid will be present. What's the color of the DCPIP? You can check here, it's blue. In case of the presence, it will become decolorized. Then DCPIP retaining its purple color, it means ascorbic acid will be absent. So this color here, we are speaking about the purple color of the DCPIP, or you speak about the blue color. It can either be purple or blue. That if any of those colors are retained, it means it will be absent. Let's go to the last one, which is the emulsion test. That is the test for the lipids. We can either use the milk or use a filter paper. So ethanol, we have the ethanol one. So add ethanol to the food sample and shake. Transfer the contents into another test tube full of water. Hope you're getting the instruction. We're saying add ethanol to the food sample and shake. Transfer those contents after adding of the ethanol, like milk. Then you transfer to another test tube full of water. So you fill it with water. The white emulsion, if it's formed, lipids will be present. So you can check at this point, it was a bit white. We are speaking about white emulsion, you can see, and it's indicating the presence of lipids. Then no emulsion formed, lipids will be present, just like this one. In case it retains this, it means lipids will be absent. You can check the diagram here. That is no emulsion. And the last one, we have the crease spot test. That is when we are testing lipids, but we're using a filter paper. So you spread that food sample on a filter paper, dry it over a flame, then observe against the light. So you're saying you're taking maybe your filter paper, you spread that food sample, you then pass it over a flame, a burning flame, observe against the light. In case a translucent mark is formed, lipids will be present. Presence of what? Lipids. If there is no any translucent mark formed, lipids will be absent. So for this food test, which are the key rules that we observe when we mark? Number one, your procedure should be very correct. And what makes your procedure to be correct? The requirements should be correct in terms of the solution required in dropwise 2 ml, 2 drops. They should be very correct. Then again, about when it comes to the solution, in solution except hydrochloric acid. Thank you very much.